All right, I think we'll get started. Thanks everybody for joining our seminar series. I'm thrilled to be able to introduce Stuart Turvey. It's a treat for us today. He ends up coming down from British Columbia, so he's at BC Children's at the Research Institute there. But in terms of the topic, how we overlap is he studies inborn errors of immunity. He's really on the leading edge of discovering genes associated with type 2 immunity, atopic disease, allergy. Um, and I ended up stumbling up on some of his work when we were doing CART11. And then I realized he's been doing this, a long time and really at Immunology ever since the beginning. He trained at, got his bachelor's and medical training at University of Sydney in Australia, and then got a doctorate in immunology at Oxford, and then went to Boston Children's for residency and fellowship, and trained with Christophe Benoit and Diane Mathis, and then has really been jumping in and solving human immune problems ever since. And so it's a real pleasure. I can't wait to hear what he's going to talk to us today. And just a reminder, please, if you're online, please mute yourself. If you have questions, put them in the chat and I'll be monitoring that. All right.
Well, thank you for that generous introduction. I'm delighted to be here. It's a pleasure to be here, to drive down the road, to come and see you, and realize, I think, how much we have in common beyond our rain-swept geography, and just I think a philosophy of focus on translational research. Before I start, I actually want to make a land acknowledgement. And so just acknowledge the First Peoples of Canada, and specifically the Musqueam, Tsleil-Waututh, and Squamish First Nations, as they were the traditional and owners of the land on which BC Children's Hospital is located, and also the University of British Columbia. And so with that, this is me. I'm a pediatrician. I see patients. And so really the research we do is embedded within this interaction. And why I emphasize that, I think, is that every interaction that happens thousands, millions of times around the world might be an opportunity to discover a brand new disease, and that we don't and that those discoveries and those patients aren't limited to, I don't know, some special ivory towers. They'll happen in every clinic. And I really encourage our trainees to be aware of the possibility that this could be something brand new, not yet described. And so when we're talking about inborn errors of immunity or immune defects, we're really talking about pediatric rare diseases. And so I think it's informative when we think about child health to appreciate how common rare diseases are in our practice. And so at a large children's hospital, it's estimated something like 1 in 3 children admitted to these large children's hospitals will have some form of rare disease. Made some good progress, but many of these rare diseases don't have a known cause. Patients and families living with rare diseases confront a diagnostic odyssey where they have test after painful and invasive test looking for a diagnosis. And sometimes they won't find a known diagnosis because that disease has not yet been described. And I think what's so exciting really is as we enter the era of precision medicine, and we start to have a molecular understanding of disease, as we start to be able to target the molecular makeup of the children. And so that is incredibly exciting for me. Let me just get my mouse here. There's a big pop-up right in front of me. There we go. When we think about medicine is driven by transformation, technological change, and we could think about many changes that are going on at the moment. AI, for example, or other changes. I guess I'd like to challenge us to think that this change is profoundly disruptive for the way we practice clinical medicine. So I started my faculty job at in the University of British Columbia in 2004, and a genome was tens of millions of dollars to sequence. So it was inaccessible, and we just didn't touch it. We didn't, and so that means that people in my generation typically don't my clinical friends typically don't really understand this technology. We weren't taught it. We're intimidated by this technology. But now here we're saying $1,000. The real price to sequence the genome is probably in the hundreds of dollars now. And so that profound shift changes our ability to sequence genomes of patients and find germline changes, sequence cancers and find somatic driver changes, sequence infectious diseases and find new pathogens. And so I think it behoves us to really embrace this technology and bring it forward to our patients. And clearly, that's a very strong theme for this hospital and for this research institute. And immunology has benefited profoundly by this change. So in 2010 is really where we date the first clinical application of whole exome sequencing. And so we can see when we look at the number of described inborn errors of immunity versus eras of time, we see a real inflection point in those 2010s where we accelerated the number of single gene defects affecting the immune system. And I think, again, what's really maybe unusual in our field is estimated that about 25% of these gene discoveries were initially described in single patients. And I think that's because we as immunologists can access the organ. We can access the blood. We can interrogate the primary cells of patients and really establish the relationship between the genotype and the phenotype. And that differs, I think, from many aspects of human genetics, where traditionally they would be gathering cohorts of 3, 5, 10 patients before they felt comfortable about describing a new human disease. And so, again, I think this just emphasizes the real value of being alive to the possibility that that patient that's confusing for us may be a brand new human disease. There's also been, I think, a big push to really embrace human immunology, and there's so many examples of that. Here's a commentary from Mark Davis back in 2008, pushing us to embrace human immunology, to recognize and appreciate mouse models, but to move beyond them. And so now I think we're realizing that we're able to harness rare human immune system disorders to give us profound insights into human biology. And while the mouse is powerful, and while the mouse may provide additional information, we don't have to rely exclusively on mouse models to advance our knowledge. And let me just sort of do a little level setting. This probably doesn't need any introduction to this audience, but what I'm predominantly talking about are inborn errors of immunity, previously known as primary immune deficiencies. So genetically determined disorders of the immune system, and when that immune system is disrupted in some way, patients present with infections, autoimmunity, malignancy. And although individually rare, when brought together, those more than 500 diseases have a relatively a high prevalence, 1 in 500 for maybe the most common, in maybe to the 1 in 10,000. And so a family doctor, an obstetrician, a psychiatrist will have patients with these disorders within their rosters. Making the diagnosis change the lives of patients. We can give accurate genetic counseling. And I think what drives many of us on the discovery side is that these discoveries of patients with single gene defects can provide insights into fundamental human biology. And we've seen many examples of that. And clearly, the local work here around FOXP3 and IPEX is a profound illustrative example of that. And the field of inborn errors of immunity really grew with a traditional emphasis of infections. And so on the left, I'm showing you this famous 10 warning signs published by the Jeffrey Modell Foundation, a patient advocacy group that said, we shouldn't try and educate the doctors about these rare diseases. We should go straight to the families and straight to the patients and suggest if they have some of these warning signs, they should go and say, hey, can you work me up for these diseases? And so there's been a strong emphasis on infection. On the right, I'm showing you images of David Vedder, the famous bubble boy born without T cells, who lived for the first 10 plus years of his life in a plastic bubble. And this is, I think, what our clinical colleagues typically are thinking about when they think about immune system disorders. But those infections really simply represent that tip of the iceberg. And now, as we've grown the list of inborn errors of immunity, we appreciate that malignancy, autoimmunity, and even more recently, allergies, so bad allergies, will also be hallmarks of inborn errors of immunity. And I think the area that we've been working quite hard in is in the area of severe allergic disease. I think it's been a little understudied, but it's also so I think sort of structurally available to us in that often clinical immunologists in weren't thinking so much of allergic disease, and so there's been really rapid growth in defining single gene defects that underlie severe allergic disease. And so my colleagues, John Lyons and Josh Milner, in 2018 coined this term primary atopic disorders. And they suggested that these are heritable genetic disorders where the major manifestations are allergic disease. And so when we look through that list of 500 plus disorders, I would say about 10% of them could likely be appropriately characterized as primary atopic disorders. And we all like mechanisms and like to put these into sort of mechanistic bins. And so those primary atopic disorders fall in different bins. Tolerance failure for IPEX, for example, skin barrier problems with diseases like Netherton syndrome, SPINK5 changes, and there's many examples. And so there are classic primary atopic disorders, hyper IgE syndrome, Job syndrome, caused by genetic changes in STAT3, or Wiskott-Aldrich syndrome, again, changes in the Wiskott-Aldrich syndrome protein that are really classic primary atopic disorders. But when I sort of talk about this and talk about the importance of thinking about these disorders, my allergy colleagues look at me, I would say, and kind of throw their hands up and say, you're describing all those patients. And we know that 10 to 20% of the population will have some form of allergic disease. Surely you don't expect me to be sequencing and working up all of those patients. And so we took up that challenge to try and identify some clinical features that'll help identify those who are more likely to have an inborn error of immunity. And it's and this is some of our attempts to do that. So when we think about these primary atopic disorders, the patients will have allergic disease. That's a given. They'll also have the clinical biomarkers of allergy or type 2 inflammation with high circulating eosinophil levels, high IgE. But beyond that, they'll also have other features that are not typical for polygenic allergic disease: autoimmunity, malignancy, growth failure, or short stature. Sometimes infections, although that's not the typical major manifestation. And then also some of the connective tissue disorders, abnormal facies, easy fractures. And, and so this is really where we've been pushing our allergy colleagues to think. They're unsatisfied by this and they want me to give them a scoring. And if I get a score of 9 out of 10 or something, we should be sequencing. It's really hard to do that. And I would suggest that actually the best positive predictor is kind of unpublishable, but it's really that an experienced clinician says, this patient is very different from the ones that I've seen in my practice. They have more severe disease. It started earlier in life. It doesn't respond. And so the message is really for people to trust themselves and recognize and see those outliers. I think the other really important message in this space is the message of broad multidisciplinary phenotyping of our patients. And so, what an allergist might see as severe allergic disease, a dermatologist might describe as severe atopic dermatitis, and a gastroenterologist might talk about eosinophilic gastrointestinal disease. And because of our sort of siloed nature of our subspecialties, we often zoom in on these elements and kind of miss the multi-organ components. And so we really are huge fans of interdisciplinary um, phenotyping, so we understand the full manifestations of these diseases. And I preach that because I've made that mistake many times. And so here's just a tiny illustrative example. We recently published a paper describing a new inborn error of immunity related to a genetic change in the gene that encodes TH-POC. And so TH-POC is a transcription factor. It's kind of famous in immunology because it's a really important transcription factor for making CD4 cells. And so, as an immunologist, we were struck by this patient's absence of CD4 cells, and we're very excited about that. But my respiratory colleague said, that's fine, Stuart. His CD4 cells are low. He doesn't really have that many infections. But the thing that's changing his life is his fibrotic lung disease. His lungs are terrible. They have this honeycombing fibrosis, and that's what you should be thinking about. And so, through that multidisciplinary um, look at things, we looked much harder at this and actually showed that TH-POC is a negative regulator of fibrosis as well. So loss of TH-POC not only affected T cell development, but the fibrosis. And that was really, I think, an underappreciated part of TH-POC biology. And again, a good reminder of how here, a single patient reminded us or informed us about human biology. So with that little introduction, I just want to take you on the journey that we've been on over the last few years, thinking about a new primary atopic disorder. And um, most of the work that I do, I think, seems to happen in the Starbucks lineup of the hospital. I think we line up there and we see our clinical colleagues and they speak to us, and many good things happen in the Starbucks lineup. And so now, several years ago, I was in the lineup and a senior clinician came and said, "Look, I know you're interested in these really rare patients who have these bad allergies. There's a one young man. He's going to have one of your things. Go and find him. He will have it for sure." Um, and practicing what I preached, we took him up on that offer. We reached out to this young man, and this was his story. He wasn't so young when we reached out to him. He was about 33 at the time, but had been one of these patients who had lived in the children's hospital, seen by many clinicians. And so he was the only child born to unrelated Caucasian Canadian parents, no notable family history. Clinicians used all the latest treatments across the years. It was very difficult to control this. As time went on, he added more and more allergic morbidity. He developed eosinophilic gastrointestinal disease, eosinophils all the way through his gastrointestinal tract, and multiple food allergies for which he had life-threatening anaphylaxis. So he ended up on a very restricted diet, essentially kind of meat and potatoes, were the only things he could successfully eat. He had bad asthma. He didn't grow well. I'll show you his growth charts. He had some infections, but these were infections that really just reflect the skin not being in such good condition. So, Staph aureus, group A strep, Candida, viral infections of the skin, but not deep organ, deep seated infections that might suggest a profound underlying host defense problem. And really, the only thing that gave him any peace at all was ongoing, quite high doses of oral corticosteroids. He could never wean the dose down very low, and not surprisingly, had many side effects from that. He had very high blood levels of eosinophils and high serum IgE. So we called him up. he consented to be involved in our research study. We sequenced his genome and his parents. When we thought we had an idea of what was going on, and you know, the lab is slow. This was probably close to a year after we'd taken those samples. We called him to share this news and to see if he wanted to help move forward with us. And the family said that he died the week before, and so at the age of 35 had died of a cerebral hemorrhage. And I'll show you that I think this too is part of the phenotype of this condition. Here's his growth charts. Pediatricians love growth charts. Here we see these some of these values below the third percentile that would meet the criteria for failure to thrive. And our approach to this, which I think is really our collective approach, how we typically do this, is we'll partner with clinicians who identify these outlier patients. Um, we might sequence them as we did in this example, or maybe more recently, as clinical sequencing has become more available, the will come to us with candidate genes. We'll go to the lab to understand how that genetic change might affect cell function. Um, we like to test therapies in vitro to see if we can identify therapies. And then we reach out to our colleagues to see if we can come together, reaching out through the listserv of the Clinical Immunology Society, Gene Matcher, as an example. In this example, we also posted a preprint and looked to identify more and more patients. And so this is the cycle through which we described STAT6 gain of function disease. So we showed how human germline heterozygous gain of function variants in STAT6 cause severe allergic disease. And I'll sort of take you through that journey in the next few slides. So one of the things I like about my job so much really is we sequence patients, genes turn up. Sometimes I know about them, STAT6, other ones I don't know about them. And then we get to study these genes based on starting with our patient phenotype. And so when STAT6 turned up at the top of the list of variants that might be possible, it was kind of a no-brainer. STAT6 is the transcription factor that lies at the heart of allergic inflammation. And it seemed a, just like a huge shock that people hadn't described this condition earlier. So STAT6 is a transcription factor that mediates the signal from those classic Th2 cytokines, IL-4 and IL-13. And IL-4 and IL-13 really lie at the heart of much of allergic inflammation, whether it's the class switching to IgE that underlies type 1 hypersensitivity, the recruitment of eosinophils, the airway remodeling and mucus production in asthma. So not surprisingly, these lie at the heart, and this is why we target them with effective medicines such as dupilumab. What was also reassuring, really, was the fact that the JAK-STAT pathway is really becoming increasingly enriched with genes that underlie human primary atopic disorders. So the classic PAD would be Job syndrome, or caused by dominant interfering changes in STAT3. Then we added on STAT5B gain of function or JAK1 gain of function. And we're starting to fill in this pathway as showing many primary atopic disorders. So we were starting to mount biological evidence that change in STAT6 was a good candidate. The other huge piece of evidence came from population genetics, where we had more than a decade of multiple genome-wide association studies that suggested that population level change in STAT6 would underlie many allergic phenotypes. And so STAT6 became, a very compelling biological target to move forward on. And so we reached out to our colleagues and quickly moved from our single patient on the west coast of Canada to a really large and delightful global cohort. And it maybe pause for a moment and might sound sort of emotionally overwrought now, but just to say that I think this is a defining feature of the global clinical immunology community, I think, is this willingness to come together and partner to improve the outcomes for our patients. And I would acknowledge Hans as one of our leading immunologists at building this spirit of collaboration. And I guess I hope that I can acknowledge and continue that spirit, because that's how we advance. And for me, this is my favorite part of my job is this partnerships, and I feel that same spirit of collaboration here. And so we're excited now to sort of be reporting and identifying patients with STAT6 gain of function on this map all the way, I guess, from Vancouver to Sydney and everywhere in between, right? Well, not almost everywhere in between. And so I'll take you through some of these first at least 10 families, just to get a sense of what this looked like. So here are some of the pedigrees. So if we look down here, I think you can see the pointer. No, I'm pointing really carefully and no one can see it. Let's try this. Whoa. Oh, that's intimidating. Here, you get me all nervous, shaking. So let's look at family J. That's too hard to do. This is showing a classic autosomal dominant pattern of inheritance. We see each affected generation with multiple sexes. And if you have sharp eyes or you're still awake, you can see that played out in family F or family C, autosomal dominant inheritance. But I want you to also look carefully and see how many of these cases were sporadic. So no family history, and the genetic change was de novo. And so we see that played out in at least half the families here. And maybe that's not shocking. And as we've sequenced more and more patients, we realize how common de novo change is explaining sporadic disease. But when I went to medical school in Sydney, they said, you ask about family history, and if they have like other people with it, that's the genetics. And I think that lives in many of our minds. But sporadic disease caused by de novo genetic change is much more common than at least I think many of us appreciated. And so that should not put us off searching for underlying genetic disorder. And these patients all pretty much looked the same as that first young man I described. Starts with the bad skin disease. They add on additional allergic morbidities. The eosinophilic gastrointestinal disease was quite bad. They didn't have the classic invasive infections we worried about. And then in the cohort, so far, we think there's probably 2 deaths attributable to this underlying defect. One, the cerebral hemorrhage I told you about, and the second from anaphylaxis. The story was anaphylaxis to nonsteroidal anti-inflammatory drugs. But this cohort is relatively new and relatively young, so we'll have a sense of that as time moves forward. Here's a little bit what they look like. This skin disease was very bad. And I think the picture kind of helps you understand that. Very chronic, lichenified, really challenging to manage skin disease. The eosinophilic esophageal disease was also very significant and seen in many of the patients. And so here are some images that we have, but our Japanese colleagues also published another paper, and you can see that this endoscopic finding here in panel A, this is the stomach of that young child. I think that would get people's attention when you were looking down there. And again, similar, very severe skin disease. This is what it looked like in terms of these biomarkers of allergic disease across the different patients. So the IgE level in the blood was very high. We use different units in different places, but the gray is the normal range, and I'm showing this on a log scale. So we can see very elevated across multiple decades of life. And then on the right, the eosinophils. So those eosinophils were high again on this log scale, gray is normal, and the dotted line represents eosinophil count of 1.5 or 1500, which is the threshold for hyper-eosinophilia. So very high, typically prolonged eosinophil counts. This is deliberately small to say that all those other things that immunologists like to do, measuring cells and all those other things, really didn't tell us anything. And so I think the message is that we should go to genetic workup very quickly. And that's the effective and efficient way to make the diagnosis. So, functional tests to try and understand things, I think, are not helpful. And genetic workup, broad genetic workup, moving quickly is the way we'll get answers for these types of patients. And so let's look at the genetic changes we found and let's look at those. So here I'm showing you the patient variants on, I'm not going to use that pointer. The patient variants are here in red, allele frequency on the x-axis, and CAD score as a in silico measure of damage um, on the y-axis. And so the patient variants were not reported in the population database NOMAD. Here I'm showing NOMAD 2, but these are also have not been reported in the expanded NOMAD 4. And they all had CAD scores that were high. We often like kind of above 20 or so for CAD scores. They were all high. And here's where they were. So I'm showing you the linear structure of the protein, and then also a three-dimensional rendering of a STAT6 homodimer with the DNA in the middle. And so mostly we see that they're concentrated within the DNA binding domain, particularly at one amino acid position, amino acid 419. And then when we look at that three-dimensional structure, it starts to make some, I think, biological sense that those amino acid changes in these missense variants were all clustered around the DNA binding interface. They weren't randomly distributed through the protein. And something that I think was very powerful for us was to compare these germline variants that we identified in the patients to somatic variants in cancer reported in the COSMIC database. And when we line those up, some things just jump off the page. So amino acid position 419 is a hotspot for us in those germline changes, but also is the hotspot for somatic changes in cancer. And I would emphasize that, in fact, all the STAT6 changes are essentially found in B-cell lymphoma. So this is B-cell lymphoma somatic changes. And then if you have sharp eyes, you'll see that many of the germline variants that we identified were also, again, recurrent somatic variants in B-cell lymphoma. So giving some, I think, hint perhaps that we're identifying gain of function variants that were also then giving a survival advantage in the concept of lymphoma. So as I tell you that story, you might be wondering, well, what happens to these patients if they have a germline variant that's known to be enriched in lymphoma? What is their lymphoma risk? And again, this is a new disease with still really quite small numbers and short follow-up. So I can't answer that with data, but I can answer it with anecdote. And so family F, the patient here, the mother, P7, was the oldest person in our cohort. She was diagnosed with B-cell lymphoma around the age of 45 or so, was treated with standard therapy, and then relapsed with more aggressive form of B-cell lymphoma about 10 years later. And she had one of these 419 variants. So for me, I think that suggests that these patients are likely at risk of B-cell lymphoma, and we should be aware of that as we treat those patients. Yeah, it was DLBCL. So it was, yeah, so as they moved on. So let's go into some of the mechanism we did to really establish that what we'd found was true. We had, I think, a nice clinical story, a nice population genetic story, but we wanted to try and probe some of the mechanism to prove that. And so we did classic and relatively simple assays. First, we started with the luciferase assay to interrogate the function of these various STAT6 variants. And so on the left-hand side, we're showing some controls. One was changing the phosphorylation site at amino acid position 641. And so that was biochemically null and was negative in our assay. I'm showing the wild type and also another population variant with and without IL-4 stimulation. And so we see that they were all relatively low. And then the patient variants here in a somewhat saturated system um, all had much higher baseline activity that went up a little bit with IL-4 stimulation, again, suggestive of a gain of function phenotype for these variants. We also went and looked at that another way here by looking at STAT6 phosphoflow, and again showed very much the same thing, that those patient variants had increased phosphorylation at baseline that then moved up with the IL-4 stimulation. If you have sharp eyes, you'll notice that one of them, the 643 change at the right-hand side or the top, didn't work in this assay, and that might be because it's right next to the 641 phosphorylation site. But we were able to show that gain of function activity with a Western blot. And so this gave us, I think, some nice evidence that the patients had gain of function, that this pro-allergic fire was burning within them. And then we turned to primary patient cells. And I think we'll be all familiar with this, the anxiety of FedEx sending samples from far away, the joy when they arrive, and the wondering what's going to happen. And so these samples would arrive. I would breathe down the neck of my PhD student saying, show me this increased signal. It has to be there. And he'd say, oh, it looked like the same as the control. And I said, oh, we'll have to do it again. Oh, sort of the same as control. And we thought about this more and talked to other people. And they said, well, that's fine. Remember, you've got to turn the immune system off. So you've got to turn it on, but you've got to turn it off. So why don't you look at the dephosphorylation rate and see if that's different? And we looked at that in different patients. I'm showing you some data here. And what it seemed to us was that the patient dephosphorylation was much slower than the matched controls. And so essentially the area under the curve of STAT6 activation was increased rather than. So it didn't exactly recapitulate what we saw in those in vitro models, but I think showed evidence of increased activity. We're also able to access patient cells and show the enhanced Th2 inflammation phenotype here using intracellular flow cytometry to measure various readouts compared to large numbers of age and sex matched healthy controls. So we had done this work. We were sharing this information back to the patient, the clinicians, and then some of the patients. And not surprisingly, they were unsatisfied. They said, that's nice, but what are you going to do for me? What can we do now we have this knowledge? And we're obviously extremely privileged in this era to have a growing armamentarium of medicines that can specifically target various immune pathways. And so as we looked through this and wondered, how can we block this classic exaggerated IL-4, IL-13, STAT6 axis, a number of obvious candidates came out. One was dupilumab, so a monoclonal antibody against the alpha chain of the IL-4 receptor, widely available, approved down into infants. That's a nice and quite easily accessible molecule for our patients. But also the growing list of JAK inhibitors were also very nice potential targets. And so we went back to the lab and back to the assays here. I'll show you the STAT6 phosphorylation. And we tested that with a variety of the genetic changes and showed that ruxolitinib, here we did tofacitinib, and also IL-4, were all able to dampen down the exaggerated IL-4 response that we saw in relation to these patient variants. And so we shared this information with our colleagues, and they were able to then access some of these medicines for their patients, and we sort of eagerly awaited what would happen. And here is images of the young man from Hong Kong. I showed you his skin originally that was terrible, and then after several years of treatment with dupilumab, his skin condition had improved dramatically, and we could measure that with various scores or simply to really look at his skin. So it worked well for his skin, but my favorite figure in this whole study is actually this one. Um, Pediatricians love growth charts, and we believe they are sort of integrators of health. And so this is that same young man with the difficult skin who was treated in Hong Kong. And you can see how profound his failure to thrive was. And so around the age of 16, the clinician started him on growth hormone. And we can really see that he had got no acceleration in his height. And then at the age of 19 or so, he started dupilumab and he grew about 15 centimeters at the age of 19 and put on a huge amount of weight actually and moved up into the normal range. I met this young man and he talks about the change in his life where his skin was so terrible he felt he couldn't go out. People stared at him, made comments. He felt terrible. And now with this medicine, he was able to live a full and active life, and he's become a huge spokesman for inborn errors of immunity in Hong Kong with Professor Yulin Lau working with him. So this, I think, is really why we do this work, is to change the lives of patients really one by one. And this young man's life was dramatically changed. Some of the patients in some jurisdictions couldn't access dupilumab, but had better access to JAK inhibitors. And so this is a publication by Safa Barris with another patient with STAT6 gain-of-function disease. And they report remarkably effective improvement with ruxolitinib. Our patient in Iran, who also couldn't access dupilumab but started ruxolitinib, had the same results. So this child, who's about 10 or 12, had such bad eosinophilic inflammation of his esophagus that every month they had to do a balloon dilatation of his esophagus so he could eat. And then it would shrink down and then dilate it. And after 2 weeks of ruxolitinib, he didn't need this balloon anymore. So I think it talks to the benefits of targeted therapy and the ability, I think, to heal and remodel if we can turn off the inflammation. And so to wrap up, I hope I think convinced you that heterozygous gain of function genetic changes or variants in STAT6 cause a new primary atopic disorder that is really severe and presents early in life, affecting essentially all body systems. I thank my colleagues here who wrote a nice editorial about this, suggesting some clinical red flags that we might focus on that suggest the importance of this condition. I think there's still a lot of work to do about the real mechanism, the biology of how changed versions of STAT6 interact with DNA, the binding, ChIP-seq style studies, and others. We don't have information about that yet, but we do know that there seems to be increased STAT6 activity and Th2 skewing. And we've been fortunate that we've been able to repurpose some approved medicines to treat this rare disease, particularly dupilumab and JAK inhibitors. But I'm really excited that there are also coming online some more specific medicines, including, for example, STAT6 degraders that might be really effective targeted oral therapies. So I will end by acknowledging the vast number of people who participated in getting this work done, but maybe more importantly, acknowledge that as we described this condition, it was clearly a disease that was ready to be discovered when it was. And so now there's been multiple groups independently describing multiple families with STAT6 gain-of-function disease. And so I don't think this is nearly as rare as we might think it will be, and it will be out there. And so I hope as you go about your days and speak to people, you'll think about the idea that there are these rare patients carrying disabling genetic changes, and we should be open to them and ready to find them, because making those diagnoses changes the lives of patients and changes them forever. So with that, I'll say thank you.
Wonderful talk. Thank you.
Okay.
Do we have questions?
Your indication that these rare mutations very often have a de novo etiology. Now, that could be because if it's lethal, that they never reproduced. So you would never find a affected offspring. And we saw this, for instance, in STAT3 dominant negative,
Mhm.
or where it was not a gain of function. On the other hand, you could also argue that gain of function would actually induce a mutation in one of the parents. And if you think about the options of make a mistake, it's very low in a female. But with spermatogonia going wild, you will usually find, I could guess, a mutation in in this spermatogonia.
Yeah.
And there is an interesting expression, permissive spermatogonia,
Yes.
which goes back about 100 years in the European literature. And we actually had a patient with a gain of function PI3 kinase delta,
Yes.
where a truck driver in a country I will
I've heard this truck driver
not
story. Yes.
name, had in every city had a woman, and he took care of them actually, and they all had offspring, and each one of them had predominantly an affected child.
Yes.
And when you looked at the sperms of this middle-aged gentleman, 30% of the
Mhm.
sperms had this particular
You're in a
mutation.
mess. Yeah.
And so I think these old uh, investigators from Europe were right talking about permissive spermatogonia.
Hmm. Yeah.
Did you have any inclination in some of your families that there was, who was the initiator of that de novo mutation? And
Yeah, we didn't try to. We didn't. So, what we had was sequencing, including kind of next generation sequencing, and then Sanger sequencing of the blood, and we didn't see, hints of some mosaicism in the parents, but we didn't sequence deeply to be,
but
and we didn't sequence anything other than blood.
did you have families where both parents were negative and they had more than one offspring?
Uh, Maybe not actually.
Because that
Yeah,
would be an indication that
yes,
that there is
yeah,
an accumulation somewhere in the germline.
yeah. That's a great question. We had families where affected parent had multiple affected children, but that doesn't answer your question about the generation before, the origin of. Yes, yeah. I don't know. We haven't looked.
Awesome talk. So I have a question about the penetrance. Like, do you see ones with any examples of these with more mild phenotypes? And I mean, I guess one would presume they would be much more common potentially given these diseases.
Yeah.
Are you looking at that?
So, I mean, the joke is that every disease is fully penetrant when you start, right? And so so the answer would be we've not seen anyone who carries one of these kind of validated gain of function variants not have a phenotype. I would say that just like polygenic allergic disease, there's a sense that this gets somewhat better as the people age. And so we had adults who had children. They still had difficult to manage allergic disease, but not fatal disease. I think we've taken I sort of think of it like a thermostat, or we're right up there. But JAK1 gain of function might be the best example where that's just been known longer, where we found these very severe ones, but now others like Dusan Begonovich and more recently the French group have now, I think, found kind of 80% turned on thermostats and shown that these genetic changes are much more common in the population, and then show a more mild gain of function phenotype, and but are associated with disease and get better with, for example, JAK inhibitors. So right now we're claiming, fully penetrant severe disease, but I think as time goes on, we'll find the next layer down.
If it depends on or like ligand being there, which it seems like,
Yeah.
then you'd expect it to be mixed penetrance.
Yes. Yeah.
With mild.
Yeah, I think so. So I think it's just a matter of time that we'll find those ones.
Similarly, I'll jump in here. In terms of the JAK inhibitors, it's known that some bone marrow fibrosis or alopecia, when you stop your JAK inhibitor, you can get some rebound.
Yes.
So can you comment on like the feedback regulatory pathways? Like, is that what's happening as the patients age and get a little bit milder disease?
Yeah, I don't know. I mean, we've in the last couple of weeks have been kind of reassembling our global cohort to try and actually answer this question properly, which is now we have more, what's the natural history? Let's understand that it's anecdotal that the mom of the child in Iran was a little less bad than her child was, but remembers that she was terrible when she was a child. So it's clinical anecdote.
But
right
do you
now.
see when patients like miss a dose or they stop their medicine, you get like a
Yeah.
really profound rebound?
I'm not sure. I'm not sure it's above like how bad they
Okay.
were. They certainly, short half-life for JAK inhibitors, and so miss one dose, they start getting itchy and unhappy very quickly. I'm not sure if it's above the baseline.
Sure, you have enough patients, but do you see any difference in the phenotype between females and males?
I don't. We haven't looked properly. I can't answer. Sorry.
Any other questions? All right.
Perfect.
Let's thank Stuart for coming.
Thank you.